T cells play a crucial role in our immune system, and their activation requires a complex biochemical pathway. When T cells encounter an antigen, they need the help of co-stimulatory molecules to become activated. Once activated, T cells can divide and differentiate into effector cells that carry out various functions to combat the infection. In this report, we will explore the biochemical pathways involved in T cell activation and how T cells respond to antigens and co-stimulatory molecules to perform their functions. In this report, we will explore the intricate biochemical pathways involved in the activation of T cells and the functional responses of these cells to antigen and co-stimulation. We will also go through the secretion of cytokines and expression of cytokine receptors, which play critical roles in coordinating the immune response. We will also examine the process of clonal expansion and the differentiation of naive T cells into effector cells, as well as the development of memory T lymphocytes. By the end of this report, we hope to provide a comprehensive understanding of the mechanisms behind T cell activation and function, which are essential for our immune system's ability to fight infections and diseases.
Following the recognition of antigens and co-stimulators, T cells express proteins that are involved in their proliferation, differentiation, and effector functions. Naive T cells that have not encountered antigens have a low level of protein synthesis. But within minutes of antigen recognition, new gene transcription and protein synthesis are seen in the activated T cells. These newly expressed proteins mediate many of subsequent responses of the T cells. In addition, antigen recognition also activates several biochemical mechanisms that lead to T cell responses, including the activation of enzymes such as kinases, recruitment of adapter proteins, and production of active transcription factors. The figure presented shows the proteins produced by antigen-stimulated T cells. The antigen recognition by T cells results in the synthesis and expression of a variety of proteins, for example, which are shown in the figure on the screen. The kinetics of production of these proteins are approximation and may vary in different T cells and with different types of stimuli. The possible effects of co-stimulation and the patterns or kinetics of gene expression are not shown. The function of some of the surface proteins expressed on activated T cells are shown in Part B. CD69 is a marker of T cell activation involved in cell migration. The interleukin-2 receptor receives signals from the cytokine that promotes T cell survival and proliferation. CD40 ligand is an effector of molecules of T cells, and CTLA4 is an inhibitor of immune responses. So, to sum it up, the time after activation, what happens in the naive T cells first is the retention in the lymph node, and after that is the proliferation, and the effector function, and lastly is the control of response. Another figure here, this shows the signal transduction pathways in T lymphocytes. The antigen recognition by T cells induces early signaling events, which includes tyrosine phosphorylation of molecules of the T cell receptor, or the TCR complex, and the recruitment of adapter proteins to the site of T cell antigen recognition. These early events lead to the activation of several biochemical intermediates, which in turn activate transcription factors that stimulate transcription of genes whose products mediate the responses of the T cells. The possible effects of co-stimulation and the signaling pathways are not shown, but these signaling pathways are illustrated as independent of one another for simplicity, but may be interconnected in more complex networks. Activation protein, antigen-presenting cell, guanosine triphosphate diphosphate, immunoreceptor, tyrosine-based activation, mammalian target rapamycin, nuclear factor of activated T cells, protein kinase, isoform of phosphatidylininositol, specific phospholipase, and phosphatidylиnositol.
So now let's proceed to the signaling pathways involved in T cell receptor activation. Just for a recap, T cells play a crucial role in the adaptive immune response by recognizing and responding to specific antigens. The T cell receptor, or TCR, is the primary signaling receptor on the surface of T cells, and the TCR signaling is initiated when the TCR recognizes a foreign peptide presented by a major histocompatibility complex, or MHC molecule, on an antigen-presenting cell. There are several transmembrane signaling proteins associated with the TCR, such as CD3 and zeta chains, which both contain immunoreceptor tyrosine-based activation motifs, or ITAM. CD4 and CD8 co-receptors are important in TCR signaling as they facilitate signaling through the protein tyrosine kinase, LCK, which is attached to their cytoplasmic tails. LCK then phosphorylates tyrosine residues in the ITAMs of CD3 and zeta proteins, which are critical for TCR signaling. This leads to the activation of ZAP70 and the assembly of various adaptor proteins and enzymes near the TCR complex, mediating further signaling events. The image on the right shows the T cell receptor complex with the help of CD4 or CD8 receptors identifies peptide antigens in MHC molecules. Signals for TCR activation are passed through the CD3 co-receptor complex consisting of CD3 gamma, epsilon, delta, and zeta chains. CD4 or CD8 clustering with the TCR complex helps LCK kinase initiate signal propagation by phosphorylating ITAMs in the CD3 zeta chains. So, there are four major signaling pathways linked to zeta chain phosphorylation and ZAP70 or ZAP70. These are the calcium nuclear factor of activated T cells or NFAT pathway, the RAS and RAC MAP kinase pathways, the PKC theta NF kappa B pathway, and the PI3 kinase pathway. These signaling pathways are essential for the activation and function of T cells in response to foreign antigens. The phosphorylation of the zeta chain and recruitment of ZAP70 are necessary for the initiation of downstream signaling events and ultimately lead to T cell activation and proliferation. Moving on to nuclear factor of activated T cells, also known as NFAT or NFAT pathway. NFAT is a transcription factor that remains inactive and phosphorylated in the cytoplasm of resting T cells. NFAT activation requires an increase in cytosolic calcium ions concentration, which is achieved through the ZAP70 mediated phosphorylation and activation of phospholipase C gamma or PLC gamma. PLC gamma catalyzes the breakdown of PIP2, leading to the release of IP3, which triggers the release of calcium ions from intracellular stores. This, in turn, leads to the opening of plasma membrane calcium channels, allowing for the influx of extracellular calcium ions. The elevated calcium ions concentration activates calcineurin, which dephosphorylates NFAT, enabling it to translocate into the nucleus and activate gene expression. The immunosuppressive drug cyclosporine inhibits calcineurin's phosphatase activity and therefore suppresses NFAT-dependent cytokine production, making it useful for preventing graft rejection in organ transplantation. Moving on to RAS or RAC MAP kinase pathways, which involve a family of proteins including RAS and RAC, several adapter proteins, and a cascade of enzymes that activate mitogen-activated protein or MAP kinases. ZAP70 dependent phosphorylation and accumulation of adapter proteins at the plasma membrane initiate the pathways, leading to the recruitment and activation of RAS or RAC. The active forms of these proteins, RAS-GTP and RAC-GTP, initiate different enzyme cascades, ultimately activating distinct MAP kinases. The terminal MAP kinases, such as extracellular signal regulated kinase or ERK and C-Jun amino terminal kinase or JNK, induce the expression of C-phos and the phosphorylation of C-Jun, which combine to form the transcription factor activating protein 1 or AP1, which enhances the transcription of several T cell genes. For the PKC theta NF kappa B pathway, the T cell receptor of this signaling pathway involves a protein kinase called PKC theta, which activates a transcription factor called NF kappa B. PKC theta is activated by diacylglycerol, which is produced by the hydrolysis of membrane inositol lipids by a protein called PLC. Resting T cells have inactive NF-kappa B bound to an inhibitor called IKB, but TCR-induced signals downstream of PKC theta activate a kinase that destroys inhibitor kappa B and releases NF-kappa B, which promotes gene transcription in the nucleus. Moreover, this pathway plays a crucial role in T cell activation and immune response. Lastly, the PI3 kinase pathway. In this pathway, the T cell receptor signal transduction involves PI3 kinase, which phosphorylates PIP2 to generate PIP3. PIP3 activates AKT, a serine threonine kinase that promotes the survival of antigen-stimulated T cells by stimulating the expression of anti-apoptotic proteins. The PI3 kinase or AKT pathway is activated by the TCR, CD28, and IL-2 receptors. The AKT pathway is closely linked to mTOR, a kinase that promotes protein translation and cell survival and growth. Rapamycin, a drug that inactivates mTOR, is used to treat graft rejection. Now let's move on to functional responses of T lymphocytes to antigen and co-stimulation. So when T cells recognize antigen and co-stimulators, it triggers a sequence of events that result in the expansion of antigen-specific lymphocytes and the differentiation of naive T cells into effector and memory cells. T cells secrete cytokines that affect themselves and other immune defense cells. These responses of T cells are then discussed in detail next. Secretion of cytokines and expression of cytokine receptors. Cytokines are proteins that mediate immunity and inflammation in the body, produced mainly by innate immune cells in response to infection. In adaptive immunity, cytokines are mainly secreted by CD4 T cells. Interleukin 2 or IL-2 is produced rapidly after activation of CD4 T cells and increases the expression of the high-affinity IL-2 receptor. IL-2 stimulates the survival and proliferation of T cells, thus increasing the number of antigen-specific T cells. While CD8 T cells do not produce large amounts of IL-2, they still proliferate during immune responses, possibly due to antigen recognition and co-stimulation or with the help of CD4 T cells. So the image on the right depicts the function of interleukin 2 or IL-2 and IL-2 receptors in T cell proliferation. Initially, naive T cells have a low affinity IL-2 receptor complex made up of beta and gamma chains. When activated by antigen recognition and co-stimulation, the T cells produce IL-2 and express the alpha of the IL-2 receptor or CD25, which combines with the beta and gamma chains to create a high affinity IL-2 receptor. IL-2 binding with this receptor leads to T cell proliferation and aids in antigen recognition.
So let's proceed to the clonal expansion. T lymphocytes activated by antigen and costimulation begin to proliferate within 1 or 2 days, resulting in expansion of antigen specific clones in this image here. The expansion and the decline of T cell responses, the numbers of CD4 plus and CD8 plus T cells specific for various antigens, and the clonal expansion and contraction during immune responses are illustrated. The numbers are approximations based on studies of model microbial and other antigens in inbred mice. And in humans, the numbers of lymphocytes are about 1,000-fold greater. The figure evaluates the expansion that quickly provides a large pool of antigen specific lymphocytes from which effector cells can be generated to combat infection. The magnitude of the clonal expansion is remarkable, especially for CD8 plus T cells with an estimated doubling time of 6 hours. This means that the numbers of cells in antigen specific clones have increased by more than 10,000 fold. Several features of this clonal expansion are surprising, such as the lack of a detectable increase in bystander cells that do not recognize the microbe, and the majority of the expanded clones being specific for only a few immunodominant peptides of the microbe. CD4 plus T cells appear to be 100-fold or 1,000-fold less than CD8 plus cells, which may be due to differences in the function of two types of T cells. The CD8 plus CTLs are effector cells that kill infected cells by direct contact, while the CD4 plus effector cells secrete cytokines that activate many other effector cells. A small number of CTLs may be sufficient to kill large numbers of infected cells. This process of differentiation is the result of changes in gene expression, such as the activation of genes encoding cytokines or cytotoxic proteins. It begins in concert with clonal expansion, and differentiate effector cells appear within 3 or 4 days after the exposure to microbes. Effector cells of the CD4 plus lineage acquire the capacity to produce different sets of cytokines. The subsets of T cells that are distinguished by their cytokine profiles are named Th1, Th2, and Th17. So many of these cells, this image is the development of effector CD4 plus T cells. And many of these cells leave the peripheral lymphoid organs and migrate to sites of infection to where their cytokines recruit other leukocytes that destroy the infectious agents. So when naive CD4 plus T cells are activated in secondary lymphoid organs, they proliferate and differentiate into effector cells, which are the Th1 cell, the Th2 cell, and the Th17 cell. And they mostly exit the lymphoid organs and function to eradicate microbes in peripheral tissues. Other differentiated cells called follicular helper T cells remain in the lymphoid organ and help B cells to produce potent antibodies. Next is the roles of CD40L and cytokines in effector functions of CD4 plus helper T cells. CD4 plus T cells that have differentiated into effector cells expresses CD40L and secrete cytokines. CD40L binds to CD40 on macrophages or B lymphocytes, and cytokines bind to the receptors on the same cells. So the combination of signals delivered by CD40 and cytokine receptors activates macrophages in cell mediated immunity and also activates the B cells to produce high affinity isotope switch antibodies in humoral immune responses. So the CD4 plus helper T cells activate phagocytes and B lymphocytes through the action of plasma membrane proteins and by secreted cytokines. The most important cell surface protein involved in the effector function of CD4 plus T cells is CD40 ligand, and member which is the member of a large family of proteins structurally related to the cytokine tumor necrosis factor or TNF. Next is the development of memory T lymphocytes. A fraction of antigen activated T lymphocytes differentiates into long lived memory cells. These cells are a pool of lymphocytes that are induced by microbes and waiting for the infection to return. We do not know that what factors determine whether the progeny of antigen stimulated lymphocytes will differentiate into effector cells or maybe memory cells. Memory cells have several important characteristics. One of which is memory cells survive even after the infection is eradicated and antigen is no longer present. And certain cytokines, including IL-7 and IL-15, which are produced by stromal cells in tissues, and they may serve to keep memory cells alive and cycling slowly. Second is the memory T cells, and they may be rapidly induced to produce cytokines or kill infected cells on encountering the antigen that they recognize. These cells do not perform any effector functions until they encounter antigen, but once activated, they respond much more vigorously and rapidly than do naive lymphocytes. Lastly, the memory T cells can be found in lymphoid organs and in various peripheral tissues, especially the mucosa, the skin, and the circulation. They can be distinguished from naive and effector cells by several criteria. So that would be all. Thank you. And God bless, guys.